Welcome back to Cancer as a Mitochondrial Metabolic Disease Basics. Many people know mebendazole as a pharmaceutical medication for parasites. But one of the most interesting things about mebendazole has nothing to do with parasites or even microtubule disruption. What if the most critical part of its anti-cancer activity comes from its ability to interfere with cancer's fuel supply? Researchers have identified its effects on HIF-1 alpha, MYC, glucose metabolism, and the Warburg effect, some of the very pathways that help tumors grow, adapt, and survive. For someone like me who primarily views cancer through the lens of mitochondrial dysfunction and as a metabolic disease, that's far more interesting. So in today's video, we're going to ask a simple question. Can mebendazole help disrupt cancer's glucose metabolism? And if so, how does it do it? We will soon find out. So without further ado, let's get into it. So although it's important to look at a particular drug and or nutraceutical flavonoid compound as a general overview, for me, it's much more interesting to look at a compound or a drug from how it affects metabolic networks. And so I always try to get us back to this very classic Seafried graphic, which is looking at the two major biologic metabolic targets, glucose and glutamine metabolism. Today, I'm going to focus us on the left side of the graphic, which is glucose metabolism, and then we will look at, in the next video, glutamine metabolism as well. So I'm going to start out with this particular paper that was published in February of 2023, and it was titled, Mebendazole Treatment Disrupts the Transcriptional Activity of Hypoxia-Inducible Factors 1 and 2 in Breast Cancer Cells. And it says here in the abstract, mebendazole has been demonstrated to have preclinical efficacy across multiple cancers, including glioblastoma multiforme, medulloblastoma, colon, breast, pancreatic, and thyroid cancers. It later says that in addition to tubulin inhibitors, the gene expression profile that correlated most with mebendazole treatment matched the profile of cells treated with known hypoxia-inducible factor HIF-1 alpha and 2 alpha inhibitors. The HIF pathway is the main driver of the cellular response to hypoxia or low oxygen, which occurs in solid tumors. Preclinical data support using HIF inhibitors in combination with standard of care to treat solid tumors. And what they did was they looked at two kind of molecular assays to look at if the hypoxia-inducible factors were being affected at varying concentrations of mebendazole. And in each kind of target assay, they looked at the cells under normoxia or normal oxygen and hypoxia or low oxygen. And that is much more likely to be the reality at the level of a tumor in the body. So let's focus on the right part of each graphic. This is going to be looking at lactate dehydrogenase A expression, and this is going to be looking at the hypoxia response element, the HRE. And so we see that at 0.5 micromoles of mebendazole, when added to these breast cancer cells, you see a roughly 38% decrease in lactate dehydrogenase A expression, and you see a subsequent 53% decrease at 1 micromole of mebendazole when added to these breast cancer under hypoxic conditions, which is more realistic. Then what they did was they looked at the actual entire hypoxia response element and how it was expressed in hypoxia with varying degrees of mebendazole. And we see here that at 0.5 micromoles of mebendazole, we see about a 24 to 25% decrease in expression. And we see about a 36% decrease expression at 1 micromole. And you may be asking yourself, why do we care about this hypoxia-inducible factors? How does that have to do with anything to do with cancer or metabolism? Well, this is a graphic that I like to show in multiple videos because it highlights the importance of hypoxia-inducible factors and its ability to participate in what they call metabolic reprogramming, effectively reprogramming cells from a more oxidative phosphorylation phenotype to the more glycolytic and glutamine using phenotype seen in most cancers, which is why these are cancer hallmarks. So we see here everywhere there's a black lightning bolt or a red lightning bolt, we see that those are proteins that are affected by HIF1 or HIF2 respectively. So we see here this is glucose transporters. We see all of glycolysis. We see lactate dehydrogenase A, LDHA. We see the classic glutamine transporter SLC1A5, as well as glutaminase and other downstream glutamine effects, as well as fatty acid synthesis. So basically what I'm trying to show is that when you decrease the hypoxia inducible factors and their effects on the hypoxia response element and its gene expression, you're ultimately going to see decreases in all of these proteins that are responsible for metabolic reprogramming cancer. Now, I want to then look at the next paper, which is titled Mebendazole Targets Essential Proteins in Glucose Metabolism, Leading Gastric Cancer Cells to Death. And it was published in September of 2023. And it says here in the abstract that results evidenced that antitumoral and cytotoxic mechanism of mebendazole in gastric cancer cells is related to modulation of mRNA expression of glycolytic targets, SLC2A1, which is also GLUT1, hexokinase or HK1, GAPDH, and LDHA. Moreover, in silico analysis has shown that these genes are overexpressed in gastric cancer samples. And this increase in expression is related to decreased overall survival rates. Molecular docking revealed that mebendazole modifies the protein structure, which may lead to changes in their protein function. In vitro studies have shown that mebendazole induces alterations in glucose uptake, LDH enzymatic activity, and ATP production. So what is this paper actually saying? Well, it's saying somewhat similar things to what this paper is showing. It's showing that mebendazole is disrupting molecular networks that upregulate the expression of these glycolytic enzymes. But in addition to that, it's also saying that these molecular docking studies are showing that it actually may modify the protein targets as well, not just change their expression levels, which means that it may make whatever protein's there less functional than it was before. So it may have like a dual mechanism of how it affects glycolysis, which ultimately ends up with less glucose uptake, less LDH activity, and less ATP in these cancer cells through the Warburg effect or through the cytosolic substrate level phosphorylation. It then looked at each of those targets that it talked about in that abstract: GLUT1, hexokinase, GAPDH or glyceraldehyde 3-phosphate dehydrogenase, and lactate dehydrogenase, LDHA. And it looked at the expression at only 0.1 micromoles of mebendazole. And you see giant decreases in expression in GLUT1, hexokinase, less but still significant, about 50% decrease in this glyceraldehyde 3-phosphate dehydrogenase, and a huge amount of decreased expression in LDHA or lactate dehydrogenase. Now, the question is, what are estimated serum concentrations of mebendazole at what I would consider therapeutic dosages? Now, there are people out there who are recommending that people take 100 milligrams, 200 milligrams of mebendazole, 500 milligrams of mebendazole. And there is evidence to suggest, and we'll look at this, that that could be helpful. But if you look at most of the preclinical or clinical studies on mebendazole, they use somewhere between 25 and 200 milligrams per kilogram. So let's take a look at what those estimated micromolar quantities in the bloodstream would be after those respective doses. So we see here at what I would consider a minimum dose at 25 milligrams per kilogram, you're seeing about a 0.75 micromolar blood level. And at 50 milligrams per kilogram, you'll be looking at a greater than 1 micromolar blood level of mebendazole. And if you look at these studies, they're mostly using, and you'll see in other studies when we look at glutamine, that most of these studies are using 0.5 or 1 micromole as the dose that they're giving these cell cultures. And we see a 38%, a 53% reduction in activity here, 24% and 36% respectively of the hypoxia response element. And we see huge decreases in expression in this particular gastric cancer cell model with using just 0.1 micromoles of mebendazole. And we'll further look at what concentrations were used to decrease other key signaling pathways, as well as what was used in preclinical and clinical models in future videos. But what I'll say is that at most of the what I would consider a therapeutic dose of mebendazole, we are hitting the concentrations that are meaningfully seen in these in vitro and preclinical in vivo models as well, which is really big because that means that we're not having to mega dose potentially this medication to get meaningful results at the level of the cell that we want. If you like videos like this, if this helps you on your journey of potentially creating a metabolic therapy plan, please like, share, and subscribe. And until next time.